Hi students. So in the last class we have seen something about the basics of proteins. So in the unit the biomolecules, and today we are going to continue the same topic. We are going to see the types of proteins. On what basis we can classify the proteins into different classes. And the first base, okay, on the basis of structure we can classify the entire protein world into three categories: fibrous proteins, globular proteins and intermediate proteins. So why we are calling this as fibrous proteins? So what do you mean by fibrous? Fiber like appearance. So they will form like a filamentous fiber like structure. So what may be the role of these fiber like proteins or fibrous proteins? We all know about this protein keratin. So where it is? Keratin is in is a kind of protein present in our hair. So the fibrous protein normally plays a structural roles in a particular living world. So the collagen of a muscle cells and the keratin of a animal being, okay, so all belongs to the particular category, the fibrous protein. Globular protein. So what do you mean by globular protein? I told you for fibrous protein there must be some specific fiber like appearance or fiber like structure over here, but in case of globular protein the protein is in the form of globules, okay, just like a globule structure. That means you cannot say some specific structure for this kind of particular protein groups, okay. So that we are calling this group of protein as globular proteins. So example for this globular protein, immunoglobulins, okay, immunoglobulins, myoglobin, hemoglobin, all belongs to the particular globular proteins. Intermediate proteins. So what do you mean by intermediate? In between these two characters, it may not be a completely fibrous structure or it may not be a complete globular structure, okay. For example, the fibrinogen. So normally it is in a form of the globular structure, okay. So it will not form a structures like this, fibers like structures, but when the function needed from the particular fibrinogen, it will form a structure called fibrins. We all know what's the role of or function of the particular fibrinogen. They are involved in the blood clotting, okay. So whenever there is a need for blood clot, the fibrinogen, the protein will convert it into fibrins and it will be now appears as fibrous protein. So this kind of proteins are belongs to the category the intermediate proteins. They can be function as both globular and fibrous protein, okay. So on basis of composition of proteins we can classify the entire protein world into two categories: simple proteins and conjugated proteins. So what do you mean by simple proteins? So what is simple and what is conjugated? So simple proteins means these proteins contains only the amino acids. As we all know, proteins are made up of amino acids, but normally the proteins contain some other groups other than the amino acids, like glucose molecules attached, lipid molecules attached, some phosphate groups attached, okay. So there are some other groups may be attached to the particular protein to function as the complete functional protein. But in case of simple proteins, there are no other additional groups being attached to the particular amino acid chain, and these kinds of particular proteins are called simple proteins. Then what is conjugated proteins? As I told you, there are proteins, so many proteins. Normally 90 to 95% of proteins are being attached with some other groups, okay, like glucose, lipids, phosphate. If the glucose is being attached to the particular protein, those proteins are called glycoproteins. If the phosphate group is attached, phosphoproteins. Lipid is attached, lipoproteins, okay. So the point you have to remember over here, conjugated proteins means the particular kinds of, or the group of proteins are attached to some other groups to function as a complete protein. See the next category of classes. On basis of the function of particular proteins, there are so many different types of functions. As I told you in the previous class, there are some thousands of proteins present in each and every cell. For example, in case of bacteria, at least 3000 to 5000 different types of proteins are present. In case of human cell, at least 30,000 proteins are present. 30,000 types of proteins are present. So on basis of function you cannot list out the number of proteins over here, but There are major classes of functional based proteins over here: structural, enzymes, transport proteins, protective proteins, pigment based proteins, contractile proteins, and hormone based proteins. Okay, for example, insulin is a kind of protein hormone. Okay, is a protein based hormone. Okay, it is being synthesized from a gene by means of proteins or enzymes, and it is a protein product. Insulin is a kind of protein, and insulin like hormones belongs to the particular category hormone proteins. Contractile proteins, those proteins involved in the contractile function, movement, contractile function. For example, myosin and actin proteins. Pigments, there are some proteins, they contains or they possess the particular pigment characters. For example, the hemoglobin, they contain the heme group, and the myoglobin. There are some proteins belongs to protective proteins. Protective proteins means they are involved in the protection of the particular living being. For example, the immunoglobulins, antibodies, and the fibrinogen. They are involved in the protection of the particular living being. Transport proteins. There are some proteins, they are involved in the transport of molecules within the particular animal or organism. For example, serum albumins, they are gonna transport molecules through the blood from one place to another place. And enzymes, there are lots of different types of enzymes are present in the living world. As I told you in the previous class, almost all the proteins, all the enzymes are proteins. All the enzymes are proteins. There is the exception, I told you, the ribozymes. Apart from ribozymes, all the enzymes are proteins. Ribozymes are made up of nucleic acids. Just remember that. Okay. And as I told you, structural proteins, the keratin, collagen, the virus coat protein. Okay. So all belongs to the structural proteins. Okay. So the point you have to remember over here, the protein world is a very big world. As I told you, whatever may be function in the any living cell, it belongs to the structural molecules, that is called the protein. Okay, without protein you cannot move any atom, not even a single atom from one place to another place. They are the working horses of the cell. So there are so many different types of functions, structures, and their roles is being elaborated over here. And the next thing we are going to see in the particular biomolecules, the carbohydrates. So the next major biomolecule we are going to see now, the carbohydrates. So what are carbohydrates? So the carbohydrates are the hydrates of carbon. So what may be the elements present in the particular carbohydrates? So as we can see easily, carbon, hydrogen, and oxygen. Okay, it is in the ratio of 1:2:1. Okay, so carbohydrates are made up of three elements: carbon, hydrogen, and oxygen. So that we are calling this as hydrates of carbon. Okay, so in the living world there are so many different types and varieties of carbohydrates available. Their functions also vary. They may be structural or they may be functional roles, and we are going to see the details later. Okay, the point you have to remember over here, we can classify the entire carbohydrates into certain categories: monosaccharides and oligosaccharides, polysaccharides. Monosaccharides, oligosaccharides, and polysaccharides. So on what basis we are classifying these things? Monosaccharide means they contains the simple sugars. Whenever you are seeing the term sugar, you have to recall it is always related with the polysaccharides or the particular carbohydrates. Okay, so simple sugars means you cannot get any other monomer or simpler unit from the simpler unit by means of any reaction from the particular monomers. That means this is the basic unit of any carbohydrate. Okay, for example, glucose, fructose, mannose. So they are all the mono sugars. They all belongs to the monosaccharides. And again we can classify the monosaccharides into some other classes based on the number of carbon. Number of carbon, we can classify the monosaccharides into some other classes. For example, triose, tetrose, pentose, hexose, heptose. So you can see it over here. Triose. Tri means 3 and 3 carbon sugars. Tetrose. Tet means 4 and 4 carbon sugars. Pentose 5 carbon sugars. Hexose 6 carbon sugars. 7 carbon sugars. Okay, so on the basis of the number of carbons present in a particular monosaccharide, we can classify them into this much of categories. 3 carbon sugars we have already seen in the particular glycolysis. In glycolysis, we have seen 3 carbon sugars, glycerol, 3 phosphate, pyruvate, okay, and tetrosis, heptulose, pentosis, ribose, and hexosis, the very common example, glucose and fructose. So there are so many different categories belongs to the particular monosaccharide on the basis of the particular number of carbons present in a sugar. And again we can classify the monosaccharides into 2 other categories based on a presence or absence of aldehyde or ketones. Okay, and those categories are aldoses and ketoses. So aldoses, the category contains the group aldehydes, and the ketoses contains the group ketones. So for aldehyde, the example, the commonest example over here, as I told you, glucose, and the ketoses, fructose. So the point you have to remember over here in the carbohydrates, there are 3 major categories: monosaccharides, oligosaccharides and polysaccharides. In the monosaccharides, we can classify them again based on the number of carbon and the presence or absence of aldoses and ketoses. Okay, so the aldoses are the monosaccharides which contains the group aldehyde, the ketoses which contains the group ketones. Okay, for aldehyde the common example is glucose. For ketoses they contains the particular fructose contains the group called ketones. And now we are going to see something about the oligosaccharides. Or what is oligosaccharides? As I told you in the previous class, anything above or anything in between 2 to 9 units belongs to oligo. So over here, for example, we all know sucrose, lactose. So these are all called disaccharides. Why we are calling this as disaccharides? Because they are made up of 2 units of sugars, or they contains 2 monomers, and each monomer is being linked with other by means of a bond. As I told in the previous class, the bond always called the glycosidic bond. Why we are calling this as glycosidic bond? Whenever you are seeing a covalent bond in between 2 sugar molecules, you have to recall the name glycosidic bond. Okay, so what happens over here? The bond formation in between 2 monomers by the reaction called condensation. And you have to remember this technical term condensation and hydrolysis. And you have to remember these 2 terms throughout the biology class. And what do you mean by condensation? Condensation means removal of water molecule. Condensation means removal of water molecule. Hydrolysis means addition of water molecule. And what is the difference between these 2? You are going to lyse something by means of you are going to add water molecule to them. By adding water molecule, you are going to lyse something, you are going to split something. That is what called hydrolysis. Okay, condensation is completely opposite to the particular process called hydrolysis. You are going to remove a water molecule by means of uniting 2 monomers. That is what happened over here. You are going to join 2 units of monomers so that you are going to get the glycosidic bond over here. So what will happen over here? Removal of water molecule. So what is the process is called? Condensation. The other name for the condensation, as I told you, dehydration. So what is dehydration or condensation reaction over here? You are going to remove the water molecule from the or by means of joining 2 units of monomers, and you are going to form a covalent bond. The bond name over here, when it is formed between 2 sugar units, that is called glycosidic bond. Okay, and what is oligosaccharides? As I told you, any polysaccharide or any saccharides, they belongs to 2 to 9 units. If it is contains 2 units to or 2 monomers to 9 units, They belongs to the category oligosaccharides. The most common example for oligosaccharides are disaccharides such as lactose and particular sucrose. And the next category we are going to see the polysaccharides. So what are polysaccharides? As I told you, anything above 10. So more than 10 units present, more than 10 units of monomers present over here, they are going to make a chain or polymers. Say some common examples. Okay, glycogen, cellulose, starch. Okay, these are all very common examples for the particular polysaccharides. So why we are calling this as polysaccharides? All these polysaccharides are made up of so many hundreds of thousands of monomeric units. They have been joined together by means of glycosidic bond. So there are two major categories of polysaccharides we are going to see: homopolysaccharide and heteropolysaccharides. So what do you mean by homopolysaccharide, and what is the example for homopolysaccharide? Homopolysaccharide example, cellulose. Why we are calling this as homopolysaccharide? Homo means same, okay, or similar. So homopolysaccharide means they are made up of same kind of monomeric unit. That means they are made up of only glucose. So glucose will form a network of structure, a chain like structure, a network of structure. They are going to form the particular structure called cellulose. Heteropolysaccharide, that means they are made up of more than one type of particular monomeric unit. They are made up of more than one type of monomeric unit. Other than glucose molecules also present in the particular heteropolymeric or heteropolysaccharides. And again we can classify the polysaccharides into some other categories: structural polysaccharides and functional polysaccharides. So why we are calling this as structural polysaccharides? Is there any example for this? As I told you, the plant cell wall. Okay, functional, we can easily call this a storage polysaccharide. Both are common. So plant cell wall. So apart from protection, there is no other role for the particular cell wall. They are giving the protection or structure for the particular plant cell. And fungi cell wall. And insects, the layer, exopolysaccharide. Insects exopolysaccharide. They are giving the protection covering for the particular insect. Okay. And these categories belongs to the particular structural polysaccharides. These polysaccharides, they are going to give the structure and the protection for the particular living organism. Storage polysaccharides, as I told you, starch, glycogen, okay, starch in plants and glycogen in animal cells. They are going to provide the storage molecules. They are going to provide the storage units of sugars for the particular reserve food. Okay, they are also called reserve food for the particular plant cell or animal cell for the future use. Okay, and these kind of polysaccharides are storage polysaccharide. And other points you have to remember in the particular carbohydrate unit, there are two other terms: reducing sugars and non-reducing sugars, or reducing end of a sugar and non-reducing end of a sugar. So normally, according to your textbook, any right side of a particular chain, right side of a particular polysaccharide is called reducing end, and left side is called non-reducing end. And technically there are some other details also we are going to see about the particular reducing and non-reducing sugars later in the classes. And next thing you have to remember about the starch. So normally the starch can bind iodine. Okay, normally starch can form helical structure and cellulose cannot form the helical structure, and you have to remember the particular detail. Starch can form the helical structure. They will form a helical structure, and so that with the helical nature it can bind or it can interact with the iodine and it will form a blue color. The starch iodine complex, you can easily observe it as a blue color compound, and the cellulose, while you are adding the iodine to the cellulose, you will not see the particular blue color. And these are the things you have to remember about the basis of the particular carbohydrates. And what are the things you have to remember? Carbohydrates are the hydrates of carbon. They are made up of carbon, hydrogen and oxygen in the ratio of 1:2:1, and they are the normally they are providing the protection or covering or the support for the particular cell structure, and most of the time they are acting as a reserve molecule or reserve storage for the food molecules within the particular living cell. For example, the glycogen in case of animal cells and starch in case of the plant cells. There are three major categories we have seen: monosaccharide, oligosaccharide and polysaccharide. In case of monosaccharide, again we are classifying the monosaccharides on the basis of number of carbon or the presence or absence of aldehyde and ketone groups. And in case of polysaccharide, as I told you, any sugar which contains more than 1 unit of the particular sugar or in between 2 to 9 units of sugar is called oligosaccharide. The examples belongs to over here sucrose and lactose, and over here the bond formed in between the two sugars are called glycosidic bond. Whenever you are seeing the term glycosidic bond, you have to recall the carbohydrates. And next category we have seen the polysaccharides, and there are so many categories we have seen in the polysaccharides. Among the polysaccharide, the structural polysaccharide and storage polysaccharide, homopolysaccharide, heteropolysaccharide, and finally we have seen something about the helical nature of the starch, so that it can bind with iodine, so that it will form the blue color when you are adding the iodine to the particular starch. And now we are going to see the next category of biomolecules called nucleic acids. And now we are going to see the next category of biomolecules, so the nucleic acids. As we all know, there are two major kinds of nucleic acids present in any living world. It may be DNA or it may be RNA. So DNA refers to deoxyribonucleic acid and RNA refers to ribonucleic acids. Okay, and what are the basic components present in a particular nucleic acid, and what is the function of the particular nucleic acid? So the nucleic acids are the genetic material. They are the genetic materials of any living cell. Okay, it may be a virus, it may be a bacteria, it may be a fungi, it may be a plant cell, it may be a human cell. Whatever may be the kind of cell, it is made up of it contains the information is in the form of DNA or RNA. Okay, I told you RNA. Most of the cells, 99% of the cell which contains the genetic information is in the form of DNA, and some viruses, RNA viruses, which contains the information is in the form of RNA. Okay, so that we are calling these groups DNA and RNA as genetic material of cell. So they are carrying the information for the function of the particular cell. The complete information about the particular cell is being stored in the particular nucleic acids. And what are the things you have to remember about the nucleic acids, or what are the components of nucleic acids, and what are the things present in the particular nucleic acid? And you have to remember the term nucleoside, nucleotides, nitrogenous bases, ribose, sugar, phosphoric acid or phosphate group. And these are the terms you have to remember while you are studying the nucleic acids. Okay, and we are going to see one by one. So whatever may be the nucleic acid, it is made up of some basic unit. The basic unit present in the particular nucleic acids are the nitrogenous bases. And what are the nitrogenous bases present in the particular nucleic acid? Adenine, guanine, thymine, cytosine and uracil. And these are the nitrogenous bases present in the particular nucleic acid. It may be a DNA or it may be RNA. It is made up of this combination of the nitrogenous bases: adenine, guanine, thymine, cytosine and uracil. And whatever may be the nucleic acid, it contains the ribose sugar. Okay, it may be a just ribose sugar or it may be a deoxyribose sugar. Okay, it may be a ribose or deoxyribose sugar. Simply you can say a pentose sugar is present in a particular monomeric unit of the particular nucleic acid. Okay, a pentose sugar. Okay, so nitrogenous bases plus a pentose sugar, they are gonna together they are gonna make the structure called nucleoside. So what is nucleoside? Whenever you are seeing the term nucleoside, you have to say it contains two groups, a pentose sugar and a nitrogenous base. For example, over here If it is a DNA, just imagine this is a nitrogenous base adenine along with the sugar deoxyribose. It will make a adenosine. Adenosine is the name for the particular nucleoside. Adenosine. If it is guanine, we can call this as guanidine. Thymine, thymidine, cytidine, urididine. Okay. So the point you have to remember over here, whenever you are combining the particular nitrogenous base with the ribose sugar, you are getting the nucleoside. Don't get confused over here. Nucleoside refers to the absence of the phosphoric acid or phosphate group. Then what is nucleotide? As I told you, nucleotide means plus a phosphate group. Okay, that means that particular nitrogenous base, the pentose sugar combination is being linked with some other nucleoside. Okay, so nucleoside, there are more than one nucleoside is being attached to each other by means of a phosphoric acid bond, phosphodiester bond. We have to say phosphodiester bond. Okay, it's a kind of covalent bond formed in between the phosphoric acid groups. Okay, as we have seen in the ATP molecule over here also, the phosphodiester bond, that is what gonna form in between the two nucleosides. So whenever you are seeing the term phosphoric acid is present with the nucleoside, that term is called nucleotide. So the point you have to remember over here in the nucleic acids, there are two types of nucleic acids, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Why we are calling this as a deoxyribonucleic acid? Because in case of DNA, the ribose sugar is deoxyribose. In case of DNA, it is deoxyribose. In case of RNA, it is just a ribose. And other terms you have to remember over here, the nucleosides, nucleotides and the nitrogenous bases and the bonds in between the nitrogenous base and the particular sugar. I told you the nucleoside, in the nucleoside it will make a bond in between nitrogenous base with the particular sugar. And what may be the bond over here? The name of the bond, as I told you, whenever you are seeing the term sugar, if the sugar is going to make a bond with something, that bond must be called glycosidic bond. And next thing, as I told you, nucleotides. So over here, these nucleosides are combined with each other by means of the phosphodiester bond. Okay, and these are the terms you have to remember for the particular nucleic acids. And now we are going to see something about the structure of the particular nucleic acids, the DNA. So the final structure of DNA is being elucidated by the Watson-Crick. So the point you have to remember over here, the DNA is normally a two-dimensional structure of DNA. Is a double helical nature. So normally it is made up of two polynucleotides. What is polynucleotide? Any nucleic acid is made up of nucleotides, and when you are combining the nucleotides, and it will make a polymeric structure, and this polymeric structure is called polynucleotide. As I told you, it is a double helical nature. That means double. There are two polynucleotides are present over here. Okay, so they will form a helical like structure over here, and within a helical like structure, and each helical like structure and each strand contains the bases, nitrogenous bases, and each nitrogenous base gonna form a complementary base pair. Complementary base pair. So I will tell you the base pairing: adenine, guanine, thymine and cytosine. Okay, adenine always pairs with thymine, guanine always pairs with cytosine. Okay, so over here you can see there are two hydrogen bonds always present. Adenine pairs with thymine with two hydrogen bonds, and guanine pairs with cytosine with three hydrogen bonds. So this is what called Watson-Crick base pairing. Okay, so the point you have to remember over here, adenine always pairs with thymine, guanine always pairs with cytosine. Okay, in a particular helical structure you can see the base pairing or hydrogen pairing in between these two chains. Okay, so it will form a helical structure. Okay, the width will be around 20 Armstrong, the height will be around 34 Armstrong, and the distance between two bases will be around 3.4 Armstrong. We are going to see the detailed structures later while we are discussing the questions. Okay, so the point you have to remember over here, whenever you are studying the DNA and its structure, you have to remember it is made up of polynucleotide chains. There are two polynucleotide chains over here, and those two chains are twisted around each other and they are going to form the double helical structure. They are running in an opposite direction. That is what you have to remember. And we can mark the particular direction as 5 prime to 3 prime. 5 prime to 3 prime. If one chain runs in a 5 prime to 3 prime, the other chain runs from in the opposite direction. If one chain runs from 5 prime to 3 prime, the other chains opposite in a opposite direction, so that it will form a helical structure. Okay, so that is what called the nature called anti-parallel nature of polynucleotides in the DNA. What happens over here? There are two chains, they are running in a anti-parallel, they are running in a opposite direction, so that they will form a helical structure. So while it form a helical structure, as I told you, among the two bases, the bases present in the opposite chains, they will form a hydrogen bonds in between them, so that it will form a stable helical structure. And other stabilizing structures present over here, the base stacking. What is base stacking? What is stacking? The arrangement one above the other. Like in the staircase, like we have seen in our home. Staircase arrangement, one step above the other. The equal space is one step above the other arrangement. Like that, you can see the stacked arrangement of bases in a particular DNA, so that it will have some stable 2-dimensional and 3-dimensional structure. Okay, and other structures that are stabilizing the particular DNA, the hydrophobic nature. The hydrophobic nature in the water environment also going to stabilize the structure of the DNA. Okay, so while you are studying the DNA, what are the things you have to remember? Their structure, their basic components, the nucleotides, and they are made up of polynucleotides. There are two chains, they are running in an opposite direction, 5 prime to 3 prime opposite direction, and the hydrogen bond in between the base pairing. And what are the base pairing? Adenine with the thymine by 2 hydrogen bonds, guanine with the cytosine by 3 hydrogen bonds, and these are the things you have to remember for the DNA. And what is RNA? As I told you, RNA is a ribonucleic acid. RNA is a ribonucleic acid, and how it differs from the DNA? Here the ribose sugar is present. Here the pentose sugar over here is ribose sugar. And other major difference from the particular DNA molecule over here is the presence of uracil. So instead of thymine over here, the uracil is present in case of the particular RNA molecule. And this is what you have to remember for the RNA. And there are so many different types of RNA and so many different types of functional roles played by the particular RNA. And the major categories of RNA we all know: mRNA, tRNA, rRNA. And what may be the role of these RNA molecules? As we all know, in a primitive stage of the biological living world, the RNAs are the genetic material. So what it means? RNAs are the genetic material. They played the role of the DNA in a primitive cell. Okay, only in between time the DNA evolved as a molecule, as a genetic material, and before the evolution of DNA, only the RNA molecule is present, and the RNA molecule itself, they have played the particular role of genetic material. So now in a present day cell, the RNA plays the role as a intermediate molecule. Why we are calling this as intermediate molecule? And now we will recall the central dogma of molecular biology: DNA, RNA, proteins. So as I told you, the proteins are the working horses of a cell. They are going to perform all the functions of a cell. And DNA, as I told you, contains the information. Any information is being needed by the cell is being decoded from the DNA. For example, if you want to synthesize, if your cell want to synthesize a particular protein, that information is in the DNA. But the point here to remember over here, the RNA molecules are intermediate molecule. Why we are calling this as intermediate molecule? The information from the DNA is being decoded into RNA, and decoded information will synthesize as the final molecule, the working molecules of cells, proteins. So from the DNA, the synthesis of RNA is called transcription. From the RNA, the synthesis of proteins are called translation. The point you have to remember over here, the DNA synthesis, replication. You are not supposed to confuse with these three terms. What is the replication? The replication is synthesis of DNA, multiplication of DNA, multiplication of the copies of DNA is called replication. What is transcription? The transcription is you are going to copy the information from the DNA. Suppose you have a original mark sheet with you. Okay, you need a copies from the particular original mark sheet. You are not supposed to submit originals to anybody. You have to keep your originals with you, and you have to copy the originals so that you can give the Xerox copies to some other purposes. Okay, so what you will do, your originals are called the DNA. What you are going to do with the RNA, you are going to make the copies, or you are going to copy the information is in the form of RNA. With the copies, you are going to use to some other functions. For example, over here, you are going to synthesize protein. So the point you have to remember over here, the RNA molecules are just intermediate molecule. They are going to link the protein synthesis with the particular information from the DNA. You have a stable DNA molecule. The DNA molecule directly will not involve in the protein synthesis. They have the information. That particular information is being copied, decoded in the form of transcription to the RNA. From the RNA, I told you there are three major categories of RNA: mRNA, tRNA and rRNA. M refers to messenger RNA. They are copying the message from the DNA. They are copying the message from the DNA so that it carries the information for the synthesis of proteins. And tRNA, T refers to transfer. They are going to transfer the units required for the protein synthesis. What are the units required for the protein synthesis? As I told you in the last class, proteins are made up of amino acids. So the tRNA is going to transfer the amino acids to the site of the protein synthesis. And rRNA, R refers to ribosomal. R refers to ribosomal, and rRNA, they are going to participate in the making of the ribosomes. They are going to make the structures called ribosomes. So together, mRNA, tRNA and rRNA, they all played in the role. They are together going to synthesize the molecules called proteins. So the point you have to remember over here, in case of nucleic acids, there are two major categories, DNA and RNA. In case of RNA, there are so many different types of RNAs. Apart from these three RNAs, mRNA, tRNA, apart from rRNA, there are so many hundreds of different types of RNAs and functions present over here, and we are going to see the details later while we are discussing the questions. And the point you have to remember over here, the RNAs are the intermediary molecule. They play the intermediate function in between the DNA and the protein synthesis. For the synthesis of proteins, the information is being copied from the DNA in the form of RNA, and they are going to synthesize the proteins. The process, you have to remember the names. Replication is called, replication means synthesis of DNA or the multiplication of DNA. Transcription, synthesis of RNA or the copying of information from the DNA. Translation, synthesis of proteins or synthesis of the polypeptides by means of the information from the RNA with the help of tRNA and rRNA molecules. In the next class, we are going to see the details of the lipid macromolecules and the enzymes and their functions.